I'm Ikari, battery number 183,847.
My name is Mushafika, battery number 181652.
My name is Daksha, and battery number is 185091.
My name is Muhammad Hafiz bin Zahari, battery number is 183512.
My name is Nishamini and my battery number is 181642.
My name is Nipinsi, battery number 18217.
Okay, today we would like to talk about what if ruminant eat too much of urine. First, I would like to give the definition of feed. Feed is material of nutritional value fed to animals. Each species has a normal diet composed of feed or feedstuff which are appropriate to its kind of alimentary tract and which are economically sensible as well as being nutritious and palatable. Agriculture animals at pasture have a diet which is very variable and subject to naturally occurring nutritional deficiencies. And then the feed grade, what is the meaning of feed grade? It's basically it's an inedible. By definition, any food deemed not fit for human consumption is defined as inedible.
Did you guys think that is weird? Like, whenever we don't eat, we give the animal, and it's like considered preservative food, although it's not normal. Okay, moving on.
I'm going to talk about non-protein energy. The portrait required for bodily function in women can be obtained by nitrogen, sources such as urea and urate. This is when the rumen microflora utilize these non-protein nitrogen sources to build their own body. When the microorganism pass through the intestinal tract of the ruminant, in particular, the bacterial protein will be absorbed and digest the same way as dietary protein. This ensures that non-protein nitrogen source can be used to supply the animal with appropriate amount of protein in a cheaper and much safer form. This could also potentially be more manageable in farm practice as this source have a better shelf life.
So you said examples of non-protein nitrogen was urine, right? So does anybody know what urine is?
Actually, let me talk about some explanations about urate. And urate is a combination of several compounds to form a set and critical options to the feed grade urea. It contains substances such as triturate, cyanuric acid, minor urea percentage, and also the urate itself. It's formed by standardized decompositions and processing of urea. As I found online, one of the methods is reheating the urea. And it contains more than 35% of nitrogen, so consequently it has the crude protein value which is 246%, which is a very high percentage. And then urate is also has a pet name which is the carbamonurea. It's an alterous compound with the molecular formula C2H5N3O2. And then talk about its physical properties. It's described that it's pelleted large crystal with high boiling point, which is 190 degrees Celsius. And then, in comparison, urates at least have 7.3 times less dangerous than urea. Low concentration of the urea in supplementations will lead to the death of animals, which compared to high level of urate in the feed. And then the chemical properties for the urate is it's unable to attract water. Its advantage in terms of burette as it has a low probability to cake and breach when its pelleted forms when compared to the urea supplementation. And what's the use for the burette? Usually we use in the agricultural product. So here's the examples. It's used as a non-protein nitrogen source in ruminant feeds where it's converted into proteins by the gut microorganism. It's less flavor than urea due to the higher cost and lower digestibility, but this characteristic also slow down its digestion and so decrease the risk of the ammonia toxicity. Do you all know that there's a test called burette test?
No.
But actually, it's called burette test, but it's not really related to the burette. It's a test. It's a test for the protein and polypeptide. It's based on the burette reagent. A blue solution will turn to valence when it contacts with the protein. And then, actually, we need is burette. It's not because actually the reagent contains burette. It's because the burette has the same response with the protein. So the burette undergo the burette test, it will change. The reagent will change into valence.
So it's to see how much protein there is. So they quantify how much protein there is on the change in the carbon. So how does nitrogen actually get metabolized?
Oh, nitrogen metabolized. Okay, so basically first we should know that nitrogen plants and animals could not live without nitrogen. It is an important part of many cells and processes such as amino acids, proteins, and even our DNA. It is also needed to make chlorophyll in plants, which plants use photosynthesis to make their food and energy. So how it is metabolized? Nitrogen is obtained from the degradation of protein or non-protein nitrogen sources in the gastrointestinal tract. So does anyone know what is non-protein nitrogen?
Yeah, I think maybe just explain.
Okay, so this starts off with the attachment of bacteria to the feed particles that are being broken down. So the bound bacteria then activates cell-bound microbial proteases to degrade and ferment the undigested feed particle. So the resulting products from this fermentation are peptides and amino acids. The rate of product formation during this fermentation process is highly dependent upon the ruminal microflora activity and the type of protein being degraded. So peptides and amino acids that extracellularly digested are transported inside microbial cells, and peptides are further degraded into amino acid. Okay. So this amino acid can then be either combined with microbial proteins or be further deaminated to volatile fatty acid, carbon dioxide, and ammonia. The fate of the absorbed nutrients will then depend on the energy availability in the body. This is a measure of the availability of carbohydrates. Excess amino acid absorbed must be excreted from the cytoplasm in the form of ammonia to prevent toxicity if it is not being used. If the body is an energized state and does not require energy from amino acid, empty amino acid will be transaminated to be used for microbial photosynthesis. If energy is limiting, the amino acid will be deaminated, forcing the carbon skeleton to be fermented into volatile fatty acid. Manipulation of the protein degradation process can be achieved by controlling proteolytic activity and also changing the peptidolysis and deamination process. Okay, so I don't know what is the fate of volatile fatty acids.
Do you guys know what is volatile fatty acids? It's a main energy source for ruminants. providing approximately 70% of the total energy requirements. They are used primarily by the microorganisms for reproduction and growth, with the excess production being used by the ruminant itself. And also, volatile fatty acids are produced in large amounts through ruminal fermentation and are of paramount importance in that they provide greater than 70% of the ruminant's energy supply. Virtually all of the acetic, propionic, and butyric acids formed in the rumen are absorbed across the ruminal epithelium, from which they are carried by ruminal veins to the portal vein and hence through the liver. Continuous removal of volatile fatty acids from the rumen is important not only for distribution but to prevent excessive and damaging drops in pH of the rumen fluid. As for the fate of volatile fatty acids, the volatile fatty acids that is being produced by the amylation process of the amino acids from the ruminal bacteria can be classified into 3 different types. First will be acetic acid. Acetic acid is utilized minimally in the liver and is oxidized throughout most of the body to generate ATP. Another important use of acetate is as the major source of acetyl-CoA for synthesis of lipids. Next will be propionic acid. It is almost completely removed from the portal blood by the liver. Within the liver, propionate serves as a major substrate for gluconeogenesis, which is absolutely critical to the ruminant because almost no glucose reaches the small intestine for absorption. Lastly will be butyric acid, most of which comes out from the rumen as the ketone beta-hydroxybutyric acid, and it is oxidized in many tissues for energy production.
Okay.
Go on. All of the volatile fatty acids appear to be absorbed by the same mechanism, which is diffusion through epithelium down a concentration gradient. As they pass through the epithelium, the different volatile fatty acids undergo different degrees of metabolism. Acetate and propionate pass through the epithelium largely unchanged, but almost all of the butyric acid is metabolized in the epithelium to beta-hydroxybutyric acid, a type of ketone bond.
So like in our case, when we were doing this report, I just thought that because like Kelly said that urea would not release ammonia slowly, and so it would not have ammonia toxicity. The reason why I figured that we should include like non-protein nitrogen metabolism and nitrogen metabolism was so we can explain that urea is part of a non-protein nitrogen, which then gets broken down. Which then gets used up to make microbial protein, and then that gets broken down as if it's a normal nitrogen, and then that goes in. But just because it is made out of urea, it releases the ammonia slowly, and hence even the byproducts slowly. So there's actually like a very fun diagram that, if possible, you can explain. But let's try to see if this works. Happy to know that. Okay. So the diagram, it's pretty small. Okay, so like all this one is actually just the nitrogen metabolism. So this dietary protein can include either normal diet protein or even your microbial proteins that come in, and then some of it that they can't digest then goes to undegradable, but the ones that can be degraded by the proteases will then be amino acid and peptides, and they will go in. Amino acid can then be used to make microbial proteins, or they can go in because peptidases get broken down to amino acids, like the peptides get broken down to amino acids, and then they make ammonia. So this process is what gets slows down when using burette. So a burette goes makes amino acid, then it goes into here, and then it makes ammonia slowly. So that's how like I made sense of how burette can get metabolized. In the case of of our cattle to see what happens to it. I just predicted that it will not suffer from toxicity, but it would still have a gain of ammonia because it is getting more ammonia than it normally does. But that would then increase physical immunogenic activities, so that would increase acetylcholine, that would increase your TCA cycle. You're getting more energy, you're getting more fat storage and all that. But at the same time, you're not getting it in a bad way to harm the body. So that's how I thought. Anything else?
So basically, how much nitrogen or the urate do you use?
I don't know how much. Normally they put it. There's like a special calculation. That's how we did. I remember I know agriculture because we did the
The square.
Yeah, then we calculate how much for the concentrate and how much. So like the urea would be the concentrate part that we write on top, and the normal feed, like you said, normal feed would be at the bottom. So then you can find out how much to put. It depends on the animal actually. Like since she said urate contains about 200% of protein, I don't think you can put a lot of urate in. But maybe just a little bit of urate plus a lot, and then it would dilute out. But it also depends on how much the animal needs. Like you can't just say, like certain animal needs more protein than the rest, right? Like, I think like eating animal would be more pregnant animal would be more. So we can't just like give them all equal amount of protein. Which is the same case for any sort of concentrated cycle. You are not supposed to give them just like that. You have to base on where they're going. And like cattle that you want them to gain a lot of muscles, like feedlot cattle and all, it would make sense that they need more protein, right? Because you want them to gain muscle faster.
So according to the diagram just now, you mentioned that the urate will slow down the production of ammonia.
It would release ammonia slower. So like this, the byproduct of any nitrogen metabolism is ammonia. Any breakdown of any protein is ammonia. So urate, if you were to use feed grade urea, it would be immediately broken down and immediately make amino acids, BFAs, carbon dioxide, ATP and all that. But the byproduct of ammonia would be high. Whereas with urate, it's not that high. The ammonia release is slower. So which means that the BFAs produced will also be slower, the ATP will also be slower. But then it means that you cannot saturate the cow. The cow will not like get a sudden splurge. Like it would take in a lot, but you will not be able to digest all of it. You will digest slowly.
So basically, we detected earlier that maybe the farmer just realized that he has been doing of urate or nitrogen in the diet.
I think he can, like this because it's going to be broken down slowly. Like, but in our case, they said he ate like 10 kg of urate. Which is like, that means he already ate it, so what happens there? But like, who would just leave the cattle to eat all those urate and like go around and do something else and come back after that and like, oh, sorry. He ate too much. Now let's figure out what happened. Yeah. But I think also after the microbial like proteins concentration, because you would get, instead of making dietary proteins, you'll be making microbial proteins. So your microbes will be more stressed to make the protein. So I don't know if that would cause some of them to die off, or if that would alter anything. Because the microbes are working harder, right? Because they're getting more source now. So I don't know if that would cause anything. Maybe I should.
The biochemistry will learn is the ammonia will enter the urea cycle.
Yeah. So I can't really remember the urea cycle. The urea cycle is like quite
No. This is the latest one. I mean,
the one with aluminum and all that.
Urea cycle is the carbohydrate.
Oh yeah, for example, is the lactate carbohydrate one. So ammonia with the aluminum trans and would affect the urea cycle?
It would, I think it definitely will. Yeah, because okay, you said it was carbonyl urea, right? Okay, let's say here they have carbonyl phosphate that starts off the urea cycle. So maybe it will increase the initiator of the urea cycle. So you get the low carbon. But it will not be as high as if you were to actually use urea. Because then I think the byproduct of this cycle is ammonia. Urea. I mean, it's the urea citrulline aspartate. It's arginine and ornithine cycle. And it happens in the mitochondria. Any other questions?
No, we're good for now.
I forgot to, I didn't get it. Actually, the burette is like mixed in the food or it's already in the food. I mean,
I don't know, it's mixed in the food. It's like a concentrate, like in a form of powder, kind of similar to urea that you mix into the food to increase the protein source.
Oh, so it's from the other sources, but naturally.
No, that's why it has like she said cyanuric acid, cyanuric and all this.
That's alright. Okay, thank you. Bye bye.